right, we're closing the doors. That means something. All right, well, welcome back to the last part of our discussion today. This is going to be fun. And I want to welcome also re-welcome actually, our scientific and research colleagues who have been having a good series of discussions over in the scientific track panel. We've been following that on the Twitter feed. We're delighted that you are part of this, and Chris Boudreau, we are really grateful to you for not only coming up with this idea, but then making it work. We had terrific participation and attendance. This is something we want to continue to do. It's a great feature and one that's long overdue for this conference, and really appreciate your leadership. One of the reasons that we are able to organize this summit, in addition to the wonderful creative energy of people like Dr. Boudreau, is because of our sponsors and our supporters. And one of our longest supporters, in fact, I believe these folks have supported this conference every year since its beginning 12 years ago, is the law firm of Faegre Baker Daniels. And I'd now like to invite Andy Campbell, who's a partner at Faegre Baker Daniels, to come up and introduce our keynote presentation this afternoon. I think he's looking forward to this because this is going to be a really cool presentation. So Andy, come on up and we'll get it started.
I was really hoping for some cool walk-on music like we had earlier, but no such luck. Well, thank you, David, and to BioCrossroads. We are excited to again partner with the Indiana Life Sciences Summit for a 6th year. Faegre Baker Daniels is a life sciences law firm, and we have a fundamental belief that the world is a better place when we can bring safe and effective drugs and medical devices to the market as quickly and as safely as possible. And to do that, we work with companies from startups, like we heard earlier in today's summit, to global leaders on all aspects of the cycle of innovation, from research and development to clinical trials to FDA approvals, marketing and promotional opportunities, to evaluating and protecting against potential risk associated with products. That is why I'm truly excited to introduce our keynote speaker this evening, Danny Cabrera, CEO of BioBots. Now, at 24, Danny is younger than I was when I started practicing law more than 10 years ago. And in just 2 years, the accomplishments that Danny and BioBots have put together is far more than what I can summarize for you in this short introduction. But let me sum it up this way. BioBots is ushering in the age of 3D biology. They're moving away from the two-dimensional face of the Petri dish in research and moving toward the three-dimensional environment of living tissue in the human body. BioBots 3D bioprinters offer the first step in the fabrication process to make that technology a reality. They are creating 3D printers that print in living human tissue. They're able to print heart and liver tissue to test the toxicity of drugs. They're printing human skin to replace traditional animal testing in cosmetics. They're exploring new materials for prosthetics and medical device implants. And they're researching cell-based therapy for more individualized and personalized medicine. I just learned that Indiana University and Purdue University have BioBot 3D printers at those universities. And I certainly cannot wait to hear from Danny about how this groundbreaking technology is being implemented in Indiana and across the world. So help me in welcoming Danny Cabrera.
Okay, good afternoon, everyone. Mic's on, good? Okay, great. Well, hey, thank you so much. That was a great introduction, Andy. He kind of covered about half my talk there, so sorry if you're going to hear some things over again. It was very thorough. But yeah, like you said, I'm Danny. I am one of the co-founders and the CEO of BioBots. And we're building tools that are empowering scientists to design and engineer living things. And why are we doing that? I mean, we really believe that biology has a huge promise. It has the promise to help us design new therapies that can help us cure diseases. We can also use biology to design and engineer new organs that can help us eliminate the organ waiting list. And even beyond that, do things like build new plants or new organisms that can help us suck the carbon dioxide out of the environment and revert global warming or climate change. And I don't know if you guys have seen Elon Musk's latest plan to colonize Mars, but biology is definitely a part of that. We need to design living things that can survive on the planet and can help us terraform it. I mean, it's a lot of stuff. You know, there's a lot of great things, a lot of great things that biology can help us achieve, can really help us do. But there have been a few things that have been holding it back, really stopping it from achieving that full potential. And the first one that I'd like to talk about is one that people in the science track might be more familiar with. And it's that most of biology today is done by hand. It's done by groups of specially trained technicians, graduate students, postdocs, and oftentimes it leads to a lot of error because you've got people running around doing things by hand. Not only that, it's a pretty antiquated model. Nothing else that we build in today's society works this way. So why are we still doing biology by hand today? The other one is, as Andy sort of mentioned, is the petri dish. I mean, we've been doing biology on flat, two-dimensional surfaces for the past 150 years. It's really since it was pioneered. And a two-dimensional surface isn't a good mimic for the complexity of the human body. So if we want to really begin to take advantage of the technology and really push it forward, we need to have better tools that let us integrate it in 3D in a much more realistic environment. Now, this isn't new stuff, but it's useful to think about what would happen if, what could happen if we could think of biology the same way we think of computers. If we really totally understood the code of life, this huge mystery of this ancient technology, what if we could understand it and rewrite it and get to a future where you can design entire living things on your laptop and print them right on your desktop? So there have been groups that have been pioneering this technology and developing it, developing useful applications from it for decades. This is Dr. Anthony Atala. He's over at Wake Forest. He's one of our scientific advisors, and he's definitely one of the pioneers in the field. Their group has developed some bioprinted, 3D printed things that have gone into people, things like pieces of bladders, kidneys, and so on. But it's even beyond just a few really high-profile researchers. In general, synthetic biologists are really used to thinking about biology in this way. They're used to comparing biology to electronic circuits. Only instead of zeros and ones shuffling across the wires, you're messing with DNA and RNA and protein, really the stuff of life. But they've really lacked the tools to take this technology outside of a few basic experiments and really distribute it all over the world. So I started BioBots with a friend of mine, thinking that we could build tools that could really get this started. And our first product was a 3D printer. Looks like this. We built them in our dorm rooms using plastic 3D printers, and we hand assembled them and basically shipped them off all over the world. But let me just explain to you how it works pretty quickly. It works like a traditional 3D printer. It has a head that's moving in 3 axes, X, Y, and Z. And inside of that head, there's a syringe, sort of like a cartridge, that's full of material. And that material is a liquid, and it's got cells inside of it. And the challenge is now, as you're pushing that material out of the syringe, how do you get it to turn from a liquid to a solid without killing all of the cells? And we developed a technology that lets us do that using, in this case, visible lights. So as the material comes out, you shine lights on it, and it goes from a liquid to a solid, and the cells, don't die, which is pretty big progress. Anyway, we developed an online community of all of our early adopters. This is just some of them up on the screen. we've really found the best scientists who are working with this technology to be the first pioneers to get them in their labs and start getting their hands dirty with them. And I'll just let me quickly tell you about some of the work that they've been able to do. This was about a year and a half ago now, so there's been a lot of progress. They were printing really complex 3D geometries, which would be pretty much impossible to fabricate by hand. So automatically, that gave them a huge advantage. And they were also beginning to characterize some pretty basic things, sort of like what happens to these materials as you print them. Do they behave differently than they would if you hadn't printed them? Also, what happens to the cells as you're printing them? How do they interact with each other? And how do they interact with the materials? And even beyond that, some of these groups have been able to build entire sort of organ systems that fit on in this little piece of space. In this case, over here on the screen, I guess, yeah, oh cool, it is on the screen. The little piece is it's actually a thrombosis model. It's a fibrosis model that was developed by Alika Demosthenes' lab at MIT, and yeah, I mean it's all made out of human cells and a gelatin material. And it models fibrosis and thrombosis of the way that it would happen inside of the body in 3D. And also, now, I guess I'll let Angela tell you guys about her work, because she's got her own video.
I'm Angela Themskalasis-Mortari. I'm a professor in the departments of pediatrics and medicine, and at the moment, we are in the 3D bioprinting facility. So the way we start is we identify what body part it is that needs to be replaced. So we need a three-dimensional digital image of it, and then that gets put into the bioprinting software. And then what we do is we create what's called BioLink that contains biological materials, and it can be with or without cells. And then that gets put into the bioprinter, and it gets printed out layer by layer, being directed by the software. It doesn't matter how complicated and how complex the three-dimensional final product is, because when you're printing in 2 dimensions, layer upon layer, that complexity becomes irrelevant. Our ultimate goal is to be able to print a body part or an organ that we can transplant into a human being. What we envision is a huge impact on the lives of patients who are on transplant waiting lists, and will actually help decrease the stress on the donor supply for organs.
So that was not a sponsored video. I mean, they did that because they wanted to attract more students to the lab, and they, by having this tool in-house, they were able to get some of the best students that wanted to work with the technology, where previously that wasn't really an option for them. And as a result, they've attracted a lot of students over to UMN, and they've got a blossoming bioprinting core right now, which is pretty exciting. So anyway, I mean, like I kind of have been alluding to, it was a really successful early launch of a really early beta product. As you can see, it was covered all over the news. I mean, a lot of different news channels. It was even put up at a design museum in Atlanta. Somebody thought it looked really cool, and we lent them a unit for a few months so they could put it up for display. And off of all that success, our team was able to raise some money. I mean, initially it was just 2 of us, and all we were running off of was basically our non-existent savings and credit cards, trying to get this business up and running. But quickly enough, we realized that we really had something here. We wanted to get a few more people involved. So we raised some money and started to grow out our team. We grew out to about a dozen people, a mixture of roboticists, material scientists, tissue engineers, and software developers. So you can see a very interdisciplinary group of people. And we started putting together all the feedback that we'd gotten from our early customers into a brand new product, something that really took the field even one step further. And that's what it looks like. It's called the BioBot 1. We launched it actually about a year ago. It was last September. We launched it at a tissue engineering conference and had great reception. So let me tell you a little bit about some of the features. I know some of you guys might be interested, and I'll keep it pretty high level. But the one thing is we kept the footprint of the device pretty small, so 12-inch by 12-inch by 12-inch. And we did that because we wanted it to be able to fit inside of a sterile hood. A lot of our customers were printing with living cells, and all of the work that they were doing was inside the hood. So being able to work with the device inside the hood, keep everything sterile, was very, very important to them. The next thing is that we added an extra extruder on there. So now you had 2 different heads that you could print with. You could start mixing different materials, different cell types, and really begin to get a lot more complexity out of your geometries. And although 2 doesn't seem like a lot, when you were used to working with just 1 and by hand, 2 is a world of difference. The next thing is we really, although part of our goal is to get rid of the petri dish, we realize that we can't do it all at once. So we still interface with standard lab equipment, and we up the precision on the device down to 10 microns. You can imagine cells are about 30 microns. A lot of our customers really cared about being able to position cells next to each other really, really precisely so that they could interrogate what was happening between them, really begin to map out those interactions in a more systematic way. The last thing is we kept our original technique for cross-linking materials. We also managed to develop a few others on top of that. Now we've got people that are cross-linking their materials with temperature, using very low temperature differences to get materials to gel very quickly, and also chemically and enzymatically. So the field has definitely expanded based on the success that our customers have been having with this. So I'm just going to walk you quickly. I think Angela did a good job in the video explaining to you how this works, but just so you guys get it, it's really not that complicated if you wanted to use this. We ship these, we build these materials. We have a huge catalog of materials now, over a dozen of them. They come in little boxes like this. Our customers can just grab them, get those little vials, and add their cells straight into there, mix them up, and put them in a syringe. And you can see it's just a standard 10 mil BD syringe. They then place that inside of the BioBot, the cartridge, and they turn over to their laptop or their computer, where they can use any traditional 3D design software, CAD, AutoCAD, SolidWorks, even things like Google SketchUp or Blender, any open source software to design their 3D geometries. They can also pull them straight from Miraz or from other online libraries. They can pull them directly from there. They then import those models into our software, which turns them into printer instructions. And then all I have to do is press print, and the printer really does take care of the rest. So just to give you a quick idea of where this is all at today, most of our customers are still scientists. These tools are being used to do basic research and really map out how the biology works. And this is really, really important if we want to get to that big ultimate goal that I mentioned way back in the beginning of the talk when I was talking about all the amazing things that biology could do. We're far away from that still. Biology is still the biggest mystery of our generation, but tools like this that really help us standardize the way we manufacture with biology, within biology, and the way we measure the data can really help us begin to create computational models that take all of this into account. And then those same models are what we're going to be able to use to design and engineer those living things. So although we're still pretty far away, I think this is a good beginning, and we're pretty excited to have gotten this far. And we've got a lot more coming. So if you're interested in learning more about that, definitely go on our website. Feel free to check us out. You can also talk to us directly on there. There are people you can chat with, like right now. If you log into the website, people will be chatting, waiting for you to chat. And otherwise, thank you guys so much for listening, and I'd love to open this up for questions. Any takers? Yeah, I think these are the mics.
So I do in vitro biochemistry, and I use automation.
Cool.
So I was just thinking about the automation. So do you dedicate one instrument per usage, or you could just do multiple things with one piece of equipment?
You can definitely do multiple things with it. You can have, the interface is those syringes, so anything that you can place into those. So you do have a limit. If you're doing really massive experiments with, liters of material, it's not really going to work. It's really meant for the earlier prototyping stages or the early proof of concept experiments, because the syringes are rather small. They're 10 mil syringes. With that said, if you want a multiplex, you can stack them up and back. So you can have lots of these running, and you only need one computer to really control all of them. So it's they sort of think about them as like a unit.
Okay, thank you.
Yep.
So great talk. I was wondering how you're going to address GMP issues. That means in particular for implantables into human bodies. That's the first question. And the second one is, have you tried to make artificial meats, basically like with the texture and the three-dimensional fibers? Because I have a customer that's asking for that right now, so it would be interesting to talk to you.
Yeah, no, absolutely. So the first question is GMP. It's a great question. I mean, we are working with a couple of our customers, really trying to iron out all those issues. It seems like it's going to be a case-by-case basis right now, since it's so early, and we only have a few people that are going in that direction. We don't have a ton of resources to dedicate to making like a universal GMP facility that anybody can like rent out. But that's something that some people are doing. It's definitely doable. It takes some modification on our end, and it takes a lot of working together with whoever's actually using the device. As far as meat, it's a great application. I don't have anybody that's actually working on it right now. I know groups that are working on meat are currently like culturing it in big vats and spinning it down and like creating layers at a time that way. And the biggest challenge seems to be the cost of cells. Cells are very, very expensive, and you need a lot of cells to create like a considerable piece of meat. And it ends up, you can maybe make a burger, but it'd be the most expensive burger you've ever eaten by many orders of magnitude. So I think the technology will have some role to play in there, but it's not super clear what it is just yet. Most of the times with meat, you don't care so much about like the bigger geometry. What you care about is more about the micro geometries. And there are some opportunities for bioprinting to play a hand in there. I don't know exactly what the future holds for that, though.
We should talk.
Very nice talk. So I take it that you have collagen, type 1 collagen as the base material maybe, and you said you cross-link the different layers. And when you do photo cross-linking, what happens to the cells? You will have singlet oxygen, etc., generated that you might be using for your binding. And so don't you think that the cells get damaged by the singlet oxygen, etc.?
Yeah, great question. I mean, the answer is, we do, one of the materials that we work with is type 1 collagen, and you can print it just straight without any photo cross-linking, so you can print it using support materials. And in that case, you don't have to worry about, if like toxicity is something that you're really concerned with, you don't have to use any of these photo cross-linkers. However, collagen by itself is sort of tricky to print. You can't get a lot of great resolution out of it, which is where the photo cross-linking comes in. And what we're finding is, the toxicity isn't from the initiators; it's really from the free radicals that are happening in the reaction. And we haven't seen in our studies a lot of toxicity with them. We're still getting like 95 to 99% viability on these cells. But not just us. Our customers who are using these. We don't really do any printing ourselves outside of characterizing these materials. Our customers, who are the ones who are developing the applications, they're the ones who are much more concerned with this stuff. And they've been really successful.
I have a question. I can talk loud. I have a question about business model. Are you just selling printers, or are you developing applications for licensing out? What's your main source of revenue?
Yes, right now we're totally a hardware company. We do just sell the printers. We've got a catalog of reagents, like I said, and we also sell those. So it's like a traditional life science tools business right now, where you sell some equipment that people pay a lot of money for up front, and then they buy the reagents as they use them. And that seems to be working right now. I don't know what the future holds again for the business. It's definitely evolving pretty quickly. So there are a lot of opportunities.
I had a quick question. It sounds like there's thousands of different opportunities for using this in research. Can you give us a couple examples of the most unusual or crazy things that you've seen?
Yeah, no, I've got a great one for that. So early on, one of our early customers was the School of Visual Arts in New York. And this is like it's a really cool place. It's not the place where you'd expect to see a 3D bioprinter, School of Art and Design. However, they also happen to have a full-fledged lab. And it's a plant lab, and they don't really do any research there. I mean, they end up doing research because they have to on the way to developing really cool art projects. And they bought one because they wanted to build a brand new plant with multiple different types of plant cells, and they were working also with fungal cells in there. And although it hasn't totally manifested itself into a fully functioning being yet, I'm really excited about the project, but it's very different than anything else that we're seeing. That's one interesting one. There is, I mean, they're all sort of unusual, right? I mean, we're like people are printing living things that they're trying to either put back into people or model what happens inside of the human body in like a cubic inch. So it still is a little bit unusual, but I think the plant one is way out left field.
DJ Nag from The Ohio State University. So clearly this is the new frontier of personalized medicine as it's coming now. How much thought have you given to sort of the ethical side of 3D printing in the bio world? And I'm sure this is not the first time you're being asked this question. And the other question related to that is the sort of the price of a fully created organ. What do you envision that price point will do for medicine in the future?
Right. I mean, so the ethics behind this is definitely something that we think about all the time, especially as a lot of people try to order these from a lot of different places, and we don't know all the people who are ordering them sometimes, and we get a little nervous about selling them. But basically, what it comes down to today, the reality is that this is still really tough work. You still need a lot of expertise. You need a lot more facilities than just a bioprinter to really be able to do anything with it. And the state of the art in the technology is building these miniature tissues. They're a few millimeters by a few millimeters by a few millimeters thick. So there's not, today there's really not a lot of damage that you could do with it. And even now it's mostly research. So as far as the ethics behind that, I mean, hey, we're really excited about being able to understand what's happening, not just inside the human body, but understand biology in general. So today we're not so concerned with what type of damage people could be doing with these, but in the future, as you were alluding to, I mean, if there is a time where these devices are producing fully functioning organs, then the ethics would become a lot more complicated. And at that point, it becomes really, really important for us to think about and really, really evaluate who we're distributing these to. Who's using them, who's allowed to use them, and what they're allowed to be used for. And it's definitely not a conversation that we're going to shape on our own. It's the entire field needs to have these conversations and figure out a course of action. That includes working with regulatory bodies to guarantee that the technology is being used in a really effective way. As for the cost of fully functioning organs, I mean, hey, I have no idea what it's going to be. I assume it'll be really expensive initially. I'm definitely not an expert in the payer system. I don't really know how all that stuff works yet. But luckily I don't have to yet, or maybe not luckily. Suffice it to say that I think it will be a good thing for a lot of people.
You may have to hire a few more people to help out with that.
Absolutely.
Thanks for the presentation.
We are hiring actually. So if anybody, if you know anybody who is looking for jobs, we have openings for pretty much anything you can think of. So thanks for the cue.
Yeah, absolutely. I'm John Losicki. I really enjoyed your presentation.
Cool.
I just had a question for you you talked about the precision with which you could lay down cells. And if you look at tissues in the human body, they're quite complex with lots of different cell types. You have neurons and vasculature and muscle tissue and that sort of thing. Can you just give us a feeling for how complex of a tissue you could generate, and what's the capability as far as different cell types that you can incorporate into a single printable material, and what's the level of complexity that you can achieve with the system?
Yeah, I mean, we're still at the early side of that. Most of the most complex tissues that we're seeing are about 4 different cell types with no more than 2 materials. And like you said, I mean, hey, that's far from what's actually going on inside of tissues in the body. But even with 2 cells, most of the times we don't actually know what's going on. So being able to investigate at that level is already providing an extra layer, an extra dimension of understanding for us. And hey, there's definitely a lot of room to grow here. We're not saying that the BioBot 1 is the ultimate answer, or or even our final attempt at this, but it's a step in the right direction.
Follow-up question as well, just something to think about for the future. Typically, I think in what was illustrated in the video, looking at broken tissues that we want to replace, but have you given thought to looking at what is my ideal tissue? So we're doing a lot of sequencing right now, trying to understand the human genome. That's the building block of, but there's many factors that contribute to that. But I wonder, is there a possibility to leverage some of the current technologies that we have with imaging to envision sort of patients' ideal normal? And then in the future, if there are issues with certain tissues, to be able to reproduce those in a perfect state rather than trying to reconstruct something that's damaged.
Yep, absolutely. I mean, and even sort of, if you take that even to one step further, what about things that we don't currently have that could be useful to us, or things that are, so there's yeah, you can, back to the ethics. Who's the ethics guy? Yeah. Okay, cool. Does anybody have any other questions? Well, then I guess I will catch you guys at the reception. Thank you.
Wow. Well, Danny, congratulations. We've been working with Reiner Fisher here for about an hour and a half, and you've already stirred his interest for the Biosciences Research Institute. We would be very interested in seeing exactly this kind of opportunity and technology at 16 Tech. It's really, really cool to hear about this and see the possibilities of it. We have, so again, thank you very much. We have one more thing that we're going to do here today, because it is quieter to do this here than if we try to do it at the reception, which we've tried to do before, and that is to announce the winners of our new venture competition for this year. And once again, the companies that came forward, the startup companies that came forward for this, I think there were 31 that filed applications, most of which we had not seen before, the BioCrossroads team had not seen before, and we typically see most things pretty early. And a really strong competition, really strong group. I want to thank my BioCrossroads colleagues, Chris Eckerly, who heads up this effort, Nora Doherty, and Brian Stemme from the Seed Fund team, who put the thing together with Chris. And I also want to thank the judges who have met this afternoon, deliberated, and come up with the winners that are about to be announced. And just to tell you who the judges have been, it's Ben Pigeon from Vision Tech, our friend Scott Atkin from IOI Partners, Matt Hall from Highland Park Ventures, Greg Coy from Taft Law, Ken Miller from Developer Town, and Nora Doherty from BioCrossroads and the managing director of the Indiana Seed Funds. An impressive jury, an impressive group, and some impressive talent that they were evaluating today. So I'd like to invite Greg Coy and Ken Miller, representing our sponsors for the competition, Taft Law and Developer Town, to come up and tell us who won.
My name is Greg Coy. I'm a partner at Taft, Stettinius, and Hollister Law Firm. My partners and I at Taft are honored to once again sponsor this new venture competition. At Taft, I focus my practice on patent and trademark law and other aspects of intellectual property primarily, but we just really enjoy representing small and startup companies in all aspects of the legal services that they need, and it's been my pleasure to act as a judge in this competition.
Well, I was just wondering on the way up, we get to pick our own walk-on music, a little personality. My name's Ken Miller. I'm a partner here in Indianapolis with Developer Town. And Developer Town assists company in developing new products, from airlines to medical devices, from precision ag to advanced manufacturing. We help them design the innovations that disrupt their industries. We're proud to be an ongoing sponsor for the BioCrossroads New Venture Competition. This year, as was mentioned, there are more than 30 Indiana companies participated, and I can speak for the rest of the judges that it is very challenging to try to review such a diverse set of companies with a lot of different technologies, management teams, but it's definitely our privilege to do so. The finalists came through a process over the last couple of weeks, several rounds of presentations and discussions, and were narrowed down to 6. And I'd like to tell you that each of them deserves some level of recognition. They all have unique competencies and are interesting in their own way. Before I turn it back over to Greg to kind of walk through the winners, I'd like to kind of describe all of the 6 finalists and give you a quick preview of who is involved with this final phase. Daylight OB, they're a medical device company improving health and safety in the labor delivery units. Their first device is used to reposition a baby during an emergency second stage cesarean. Also, we had Rubicon Agriculture. They manufacture and operate automated hydroponic growing facilities and AgroBox to provide a reliable solution for growing produce year-round. We also had Rusher Medical. They're devoted to helping millions of people with compromised lung function through a portable positive expiratory pressure therapy device. Also, we had Scioto Biosciences, developing medicines to support microbiome health within the human body through novel microparticulate platforms for delivering probiotic biofilms. That was a long one, I guess. Also, we have VinSense, or should I say VinSense, innovative software that helps wine growers make better decisions based on real-time sensed data to enhance crop uniformity, production, and profitability. And last but not least, although their name starts with an X, is Xylogenics, and they use synthetic biology, well, I'll put it in my term, they make yeast, right, to create valuable and innovative products to enable sustained chemical production and optimizing yeast for commercial processes. So you can tell that is a broad gamut. So if everyone be willing to give a round of applause for all of the 6 finalists.
The total amount of the awards being announced this evening is $60,000. A pre-venture award in the amount of $10,000 is being given to recognize a finalist that is very early in its development, but worthy of recognition for its potential. The remaining awards are for 3rd place overall in the amount of $10,000, 2nd place overall in the amount of $15,000, and 1st place overall in the amount of $25,000. And as Ken mentioned, being a judge is definitely not an easy exercise. There was much vigorous discussion, lots of differing opinions, but ultimately you cannot award 1st place to everyone, and so we had to come up with our awards that we're now going to announce. As I announce the award recipients, we ask that the representatives of those companies to please stand and be recognized, or if you're already standing, wave so we can all look and recognize you. So without further ado, the Pre-Venture Award in the amount of $10,000 goes to Rusher Medical. Please stand or wave if you're in the room. There he is. Okay. Congratulations. 3rd place, also in the amount of $10,000, goes to VinSense. Is VinSense in the room? I can't see very well. There they are. Okay. Congratulations to VinSense. 2nd place, in the amount of $15,000, is awarded to Daylight OB. Congratulations. We're now going to ask the representatives of the 1st place award winner to please join us on stage. The winner of the 2016 BioCrossroads New Venture Competition, winning an award of $25,000, is Scioto Biosciences. Let's have another round of applause for the winners of the New Venture Competition. Representatives of the finalists, I think all of the finalists are represented here, and they will be at the reception following this program. So if you're interested in learning more about their businesses, or even possibly exploring ways that you can become involved with their businesses, please find them during the reception and get to know them. A big thank you to everyone who has been involved and who has participated in the New Venture competition. It's been another big success. As David said, we've just gotten to know some exciting new companies with a lot of great prospects. And so just thanks to everyone who has been part of organizing that and been a part of putting on that competition. Thank you. David?
Okay. Well, thanks, Greg, and congratulations to Scioto Biosciences. One note for you all is that the companies that come through this competition, many of them go on to find real success and real traction. I believe we've been doing this for several years. Companies involved have come forward to raise collectively around $21 million in follow-on funding. And again, many of these are very, very early technologies. Our seed fund team continues to work with companies that come through this competition, and we often find some of our most promising candidates for investment, as do our colleagues at both Indiana University and Purdue. So this is a wrap for today. It's been a great day, varied presentations and discussions today, and gives you a sense of just how rich and diverse this culture now is in the life sciences sector here. We're going to continue that richness and diversity and culture tomorrow morning with another really terrific day. We'll be wrapping up a little after the noon hour. And we're starting at 8 a.m. We're going to hear from a really terrific keynote speaker, a real national thought leader, international thought leader in the business of life science, Dr. Gary Pisano from the Harvard Business School, who is going to have a really powerful presentation for the group and wrote the book a few years ago, Science Business, which is still today the single best book on how the challenges and the opportunities, but also just the tremendous difficulties in building businesses on the frontiers of science, but the impact that they can have. So we'll be hearing from Dr. Pisano tomorrow. We will be announcing our Watanabe Award winner, and we have a real star for you in that competition this year, as you will see. And then we're going to get a chance to hear from Rainer and to hear his vision for his work and for the work that he will be bringing to the Biosciences Research Institute. So it's going to be a tremendous morning, action-packed again. But we need to get ready for that by adjourning now to a wonderful reception here, presented by our friends and colleagues at Eli Lilly and Company and Cook Medical. Thanks to both of you. Cook Medical and Lilly do this every year, and this is really a terrific event. So people are ready. The posters are out there to take a look and take advantage of the wonderful work of our scientific colleagues who have joined us. Network, enjoy. We'll see you back here at 8 tomorrow morning. Thanks.